Good afternoon, everyone. Let's get started. I'd like to begin with prayer. Is there someone who would be willing to help us out? Please.
Father, we're grateful for this day, the weekend that we had the opportunity to listen to the prophet and awesome prayer words. Speak to the mindful of help us to remember the things that we taught in our life and put the Savior first, even when life and school and work and things try to distract us. Just forget what's most important. We just pray for the Spirit to live in our faith, to us, so with us, that we be able to learn real carefulness. We love you so very much. Say this in Christ.
Amen. Thank you very much. So today by 5 p.m., I'm looking for quizzes. There's some here at the front being turned in. I've gotten some via email. Other options in terms of turning them in, you can bring them by my office. If I'm not there for some reason, you can slide them under the door. You can take them to the chemistry department offices. I'll place it in my mailbox. We also have a drop box. So we have a display case in the east-west hallway, the main hallway of the Benson building. It's just to the west of the north. There's a large set of glass doors on the north side of the main hallway. Our display case is just to the left of that. In the middle of the display cases, there's a cabinet with slots in it, and we have some drop boxes there. So any of those will do. Just out of curiosity, has anyone taken the exam? Okay, all right, that's good. I am planning to do my office hours today well, shortly after we finish here, about 3:10. You're welcome to come with questions regarding, as part of your preparation for the exam, questions regarding current material or stuff that we've gone over earlier. Do you have questions? Yes. So it's a great question. On this display here, it has a separate option for recording. On my personal copy of Zoom, somewhere in one of the options up above, it says in the profile, I think it asked if I want to automatically record, and I said yes. And I looked for that, and I don't think it got recorded. I don't know how to make my Zoom behave better, but so sorry. I tried, but so far it hasn't worked. Yes. Yes, to my departmental address, so swgraves@chem.byu.edu. If it goes to Learning Suite, eventually I will find it, but I don't monitor email through Learning Suite very often. Other questions? All right. We are going to spend part of the day today talking about the use of antibody assays. These immunoassays, actually, they're sort of a hot topic in the defense or part of the defense against COVID is the use of these antibody assays. And so we're going to spend a little bit of time talking about them. They are an important research tool. They are an important clinical tool. As you go on to have a career in medicine, you'll be ordering antibody tests for a wide variety of different molecular targets. And so it's probably useful to spend some time discussing these. So we will do that. Please note, so in terms of how to conduct immunoassays or how to format them, you will only be required to look at the one that is in the book. Okay, there is one described in the book towards the end of chapter 5. That is the only one you really need to understand. There are several formats, some of them more complicated than others, but we're going to just limit ourselves to this one very simple ELISA assay that is described in the text. Okay. Other questions? All right. So the body has a variety of different defenses. Some of these are sort of tissue-based, cellular kinds of defenses, and some of these are what are termed humoral. These are the circulating factors or cells that tend to protect us or try to protect us against foreign pathogens, in particular viruses and viral proteins. The cells that accomplish this are the B lymphocytes. The B lymphocytes are, the B stands for bone. They are generated there. Each different B lymphocyte has its own particular antibody type on it. So they're immunoglobulins, IgG molecules, but each different cell has a particular sequence for their particular IgG. In total, there are over 100 million different B cells. Consequently, there are over 100 million different antibodies that are produced by these cells. Some of these antibodies remain on the cell surface, and some of these are sloughed into the circulation. In fact, the second most abundant protein in our blood is IgG, immunoglobulins. It represents about 2 to 2.5 grams per deciliter of this particular protein. So an enormous number of these are present always in our circulation. The antibody itself, as mentioned, will recognize different things. There is a region of 14 to 24 amino acids on the tips of these immunoglobulins, termed the hypervariable region, and they are scrambled. So each B cell will have one particular clone, one particular type of antibody, but with over 100 million different B cells, over 100 million different clones, the consequence is that any foreign protein will be recognized by at least some of these antibodies. Now these B cells are screened, and those B cells that produce antibodies that would bind and interact with our own proteins, our native proteins, are eliminated. I think this happens in the thymus. They're actually removed from the circulation. But these other 100 million B cells then are released and are able to generate all of these different types of antibodies. Consequently, if there's a foreign protein, a viral protein, then typically there are going to be a half dozen, a dozen, maybe 24 different antibodies that will recognize and bind to that particular foreign protein. When they bind to this protein, this complex of viral protein and antibody is taken up by a cell called the macrophage. It is a white blood cell, it is in the circulation, but it will destroy this complex. In addition, those B cells that have antibodies on their surface that will recognize and bind one of these foreign viral proteins will be caused to proliferate. So consequently, those particular cells that have useful antibodies will be replicated rapidly. So we begin to produce many copies of these different monoclonal antibodies that work against a particular viral protein. So you can imagine this, you catch a cold and it takes a few days before the symptoms begin to be relieved. And what's happening is you're having this proliferation of these B cells that bind, or that have antibodies that bind the viral protein. So we get a proliferation of these. When we get immunized, okay, so if you went and had your flu shot, what is happening there is that the body is deliberately exposed typically to an inactivated viral protein, not the whole of the viral protein, but portions of it that will elicit an immune response. And what happens then is the body produces more copies of those B cells that have antibodies on their surface that recognize, bind this influenza protein. Not only then do you produce large amounts of IgG that will recognize this, but there is something called memory, and the immune system then is actually able to remember or to retain antibodies that will bind and inactivate that particular viral protein. As a child, you've probably been immunized against polio. You've probably been immunized against a whole host of viral proteins, diphtheria, smallpox, so on and so forth. And in many cases, this immunity will persist throughout your life. So it is a remarkable system that once exposed to viral proteins, our body's able to respond in the short term to their presence, to eradicate them, and then it leaves this supply of these same antibodies with the intent of counteracting any future exposure to that same viral protein. Okay, any questions about that? It's a very abbreviated introduction to immunity, but it is nonetheless useful to know sort of where the antibodies are coming from and how they are produced. Okay. So this slide simply is showing you a macrophage, and it is binding the antibody antigen, antibody viral complex, engulfing it, and then undergoing a process called phagocytosis. Phago means to eat, And so that's sort of what's happening here. Okay, this is a diagram of an antibody. It doesn't look quite this tidy and neat, but it nevertheless has this general appearance of a Y. The bottom part of this, bottom part of this is termed the FC region. The C there stands for constant or crystalline. And so this part here doesn't vary within a species, okay? So the FC region for my antibodies, your antibodies will all be the same with very few exceptions. These arms extending off of the trunk are termed FAB fragments. And you will hear about antibody treatments. For example, rheumatoid arthritis is now treated with FAB fragments of monoclonal antibodies that target a protein called tumor necrosis factor alpha, which is a part of the cytokine system. These are part of the inflammatory response. Rheumatoid arthritis, you have autoantibodies. They damage tissue. It elicits this inflammatory response. But the inflammation is very problematic. It leads to swelling, calcifications, and damage to the tissue beyond the antibodies themselves. Okay, let me direct your attention up here now to these tips. This is the hypervariable region right here, in this region right here. And this is composed of about somewhere between 14 to 24 amino acids. Each B cell will have a particular sequence, but there are 100 million different sequences to this hypervariable region. So we have this ensemble, this enormous number of antibodies already there, and some of them will recognize a foreign viral protein. Okay. Let me just stay here for just a second. So, what does this? So we can see the value in having antibodies as part of our humoral immunity. They will recognize, bind, and eliminate viral proteins, viral particles, so forth. We talked about using inactivated viral protein to inoculate and immunize us. In other words, I go get a flu shot, and now I have, now my body is going to respond to what was injected into it by proliferating those B cells that have antibodies that recognize and bind this foreign protein. And in doing so, the hope is that I will have enough of the right kind of antibodies to fight off exposure to influenza for the upcoming year. Now, influenza tends to evolve. It tends to change from year to year. The rhinovirus, what we normally associate with the common cold, it changes quite rapidly. It's possible for you to get a cold more than once in a year, you know. And so it really, the virus's ability to reinvent itself, to evolve, is sort of a determinant of how long immunity will last. So the cold, rhinovirus, your exposure to it will probably produce antibodies that are effective against it, but its cousin, which emerges maybe 3 or 4 months later, is able to reinfect you with a cold. The influenza, it seems like immunity is good for about a year. For something like varicella, chickenpox, probably decades. And something like polio or smallpox, probably a lifetime. Okay. Now here's how these antibodies are exploited in terms of assays. Instead of trying to generate immunity against something per se, what happens is that a target protein, let's say that we're interested in being able to measure human insulin. Very small amounts of human insulin. Then what we could do is we could take human insulin, compound it into an injectable form, and inject an animal, let's say a sheep or a dog. And what will happen is that their insulin is sufficiently different from human insulin that this injected human insulin will elicit an immune response. They will begin to grow up large amounts of antibody that recognize and bind human insulin. What then happens is you harvest this anti-serum. You actually harvest the blood of these animals. You don't exsanguinate them, you don't bleed them to death, but you'll take off enough that leaves them healthy, but you will collect a certain amount of the blood. The blood will be enriched now in antibodies that recognize and bind human insulin. Consequently, you have a probe, if you want to think of it, you have a molecule that will bind human insulin. The interesting or the important feature of this is that these antibodies, and if you just inject insulin and collect the entire anti-serum, that is just everything that's in the blood, that is called a polyclonal antiserum. It is possible to take one of those B cells that does a particularly good job in recognizing and binding human insulin, a single clone, and produce monoclonal antibodies. You do this by fusing the B cell with a cancer cell, producing a hybridoma. And these cancer cells that now are fused with the B cell are able to endlessly produce a particular antibody that recognizes and binds insulin. So why have antibodies to carry out assays? Any ideas? Well, very simply, the antibodies have the ability to recognize a single molecule in the presence of thousands or tens of thousands of other molecules. We would call this specificity. So the antibodies have extraordinary specificity for one protein, even in the presence of something as complicated as blood, serum, plasma, or some other biological fluid. The second reason is that these assays can measure these molecules at very, very low concentrations. That is, the target molecule is at a very, very low concentration. It turns out that if you're using a technique such as UV visible absorption, you have a detection limit of about 1/10,000th of a mole. Okay, so you would need to have at least 10th millimolar of something to get an absorbance. The absorbance is usually very broad and not specific. So UV absorbance is not useful in trying to measure something at a low concentration. Something like NMR is more specific, but you have to have 10 to the -5th molar amounts of the compound to be able to measure it. The problem is that things like hormones, such as insulin, are present in nanomolar or picomolar quantities. So you think about that, you're off by perhaps a millionfold. You would have to concentrate your blood 1,000,000 times. So to be able to have any hope of measuring it using sort of standard laboratory techniques. Fluorescence does a little better, but you would still need to have micromolar amounts to be able to measure something with fluorescence. And again, fluorescence in and of itself doesn't occur for very many proteins. So not particularly useful if you're trying to measure something in the blood. So antibody assays came about in I think in the 1950s. It won the Nobel Prize. It was an extraordinary, and it still is an extraordinary improvement in our ability to measure things in the clinic, but also in the laboratory. Somebody discovers a new molecule, a new cytokine in aisle 157 or something like that, and it's the new thing. Everybody wants to study it in their particular disease entity. And so some company will actually be able to generate an antibody against that, and a few weeks or a few months after the initial publication in Science or Nature, you can buy an antibody against that. You can buy antibodies against COVID to detect it, to do research. So these processes have been improved and allow for the rapid production of these compounds. Yes? So polyclonal is when you expose an animal to a foreign protein, a viral protein, and you use the entirety of the serum as an anti-serum. So the likelihood is that there will be a dozen or 24 different antibodies that are grown up in abundance, all of which recognize that viral protein, different parts of it, so on and so forth. With a monoclonal, what you do is you actually harvest the B cells, and you find one that is particularly useful. You colonize these, and you find which has a particularly strong binding affinity for the protein of interest, and then you you grow up that hybridoma. You literally do cell culture, and you produce massive amounts of these hybridoma cells, and you collect the antibodies that they are sloughing into the surrounding medium. And you can use these then as a monoclonal antibody. It represents a single B cells product. Whereas a polyclonal, you may have a dozen, 24, maybe even more different clones involved in producing the antiserum. Okay. Yes?
How are they able to be so specific? I mean, it seems to me like kind of just making a box of Legos and saying pieces.
It is, but it turns out that because these are proteins, there is complementarity between this hypervariable region for a very few of those antibodies out of 100 million. Because you have so many different combinations, it turns out that one of those at least is going to bind to and specifically to the antigen of interest, to the viral protein. So, okay, let's talk then about this. This is from the book. This describes a very simple antibody assay. Immunoassay is the term that they will use for these. This one is called an ELISA assay. So it's an enzyme-linked immunosorbent assay. So let me just walk you through what's happening. So you take, let's say, an animal's or a person's blood or a specimen of some other kind, and you take it and you coat a test tube, a plastic tube with that material. So that creates then this layer down here at the bottom where you're coating the plastic coating. As long as you do this with an aqueous solution, those proteins present in that specimen will bind to the plastic surface. Why? What's holding them there? What? A hydration case. So hydrophobic interaction. So you will then lay down this layer of proteins and other materials that are hydrophobic on the surface. You then bring in an antibody that has been developed against a particular protein. So this is the term, the primary antibody. It is able to recognize the protein of interest and bind it tightly. So that is this yellow antibody right here. Okay, so we bring this in, we allow it to interact with the surface. The hope is that every copy of protein, of the viral protein now, will have the antibody attached to it. At that point, any excess antibody is washed away. Okay, you rinse it maybe a couple times to ensure that only those primary antibodies that are bound to an antigen on the tube remain. Then you bring in a secondary antibody. So that's this green thing here. The secondary antibody recognizes now the Fc, this constant region, of your primary antibody. Very often the primary antibody is going to be a monoclonal. And monoclonals are now produced almost exclusively from mouse B cells, these hybridomas cells. So the green thing, the green antibody, is going to not recognize viral antigen blue on the surface. It is going to recognize the Fc region of mouse IgG. So that's what it's binding to here. And then you are going to, well, this secondary antibody has a reporter function on it. It is some type of molecule capable of generating a signal. In most cases now, it is an enzyme, usually horseradish peroxidase. And what happens is that after you allow the second antibody to bind to the first, you wash away any excess secondary antibody, and then you add a solution that has the substrate for horseradish peroxidase, which is, find my cursor here. Where did you go? Oh, right. Okay. So we now add substrate. We add a solution with substrate in it. It gets converted into a product. This product usually is either is something that fluoresces, or it can be, and frequently is, a chemiluminescent compound. So it actually generates light. So now we have this, this stoichiometry. One antigen, one primary antibody, one secondary antibody. So the signal is going to be proportional to how many copies of the original viral antibodies on the surface of the tube. And as a consequence, using standards, we can generate a plot that shows signal as a function of concentration of that protein. Then we can compare our unknown to that plot, the standard plot, and we'll get a close estimate of how much of the foreign protein is present, or how much of the protein is present in the cells that we're growing in our lab, or so on and so forth. Okay, questions about this? Yes?
What's the point of the green one? Why can't you just put the recognizing thing on the?
You can, but it is an extremely labor-intensive and expensive proposition to put the signaling feature on an antibody. So rather than put it on every primary antibody that you can think about, they can generate these primary antibodies, these mouse monoclonals, for thousands of different things. And then you can use a single secondary antibody that recognizes mouse IgG to be your reporter function. So it's really just for convenience. It's not to say that you couldn't do it, couldn't add a reporter function to the primary antibody. You can, it's just that more than most people are willing to do. So, okay, any other questions? Okay. All right, we are going to begin to talk about now sugars. Okay, so we're going to talk about carbohydrates. These molecules. So we've been talking up to this point largely about amino acids and proteins. We're now going to introduce a number of other types of biomolecules. The first of these is carbohydrates. We'll talk about nucleic acids. We'll talk about another group called lipids. And then we'll come back and start talking about metabolic pathways. But carbohydrates, as you know, are a feature of our food. In most societies, it represents the most abundant source of energy in food. So most, if you think about most cultures, rice and beans, tortillas. So carbohydrates are abundant, plentiful in these diets. The US diet, it really depends on whether you're into ding dongs or Big Macs. So, you know, it's, sorry. What? Yeah. All right, so I'm gonna go through a couple slides here pretty quickly, and then we'll talk a little bit about some nomenclature, and I think that'll be about all we have time for today. Okay, so. Oh, I'm on the wrong one, sorry. Okay, so the carbohydrate is really a bit of a misnomer. But the elemental composition is carbon with sort of waters attached to it. But in reality, what we have are polyhydroxylated either aldehydes or ketones. And we'll talk a little bit about these structures. They are a major source of energy, of fuel, but they're also interesting molecules. They are found in a wide variety of other molecules, including DNA, RNA, and some energy-related molecules, ATP, NADH, FADH2. They are also found in other important biological molecules. Okay. They are between 3 and 7 carbons long. They all have a carbonyl. They either have a terminal carbonyl, an aldehyde, or they have a carbonyl on the second carbon in the carbon backbone, so a ketone. The smallest and simplest of these are glyceraldehyde, which has 2 hydroxyl groups and a terminal aldehyde, or dihydroxyacetone. So The ketone is on the central carbon of a 3-carbon chain, but you have hydroxyl groups on the other 2 carbons. So here is glyceraldehyde and dihydroxyacetone. Let me point out that the dihydroxyacetone is symmetric. It has no chiral center, but you'll notice this central carbon of the glyceraldehyde is a chiral center. It is an asymmetric center. It has 4 different constituents about this tetrahedral carbon. As a consequence, it is optically active. It is chiral. It has different stereoisomers or chemistry. If we get to something as complicated as glucose, you'll notice we still have this terminal aldehyde up here. And we have 1, 2, 3, 4 chiral centers. The last carbon has 2 hydrogens. Consequently, it has a plane of symmetry and it is not optically active. Fructose, shown over here, is a ketose. It is 6 carbons in length, as is glucose, but it has now, its number 1 carbon is not asymmetric. Again, we have these 2 hydrogens, which means there's going to be a plane of symmetry. It is not chiral. The ketone is not, doesn't have 4 different constituents. Consequently, the asymmetric carbons are these 3 right here, numbers 3, 4, 5, and again, number 6 has 2 hydrogens. A plane of symmetry is not chiral. As a consequence, there are going to be different numbers of stereoisomers for these 2 types of hexoses. Okay, they're both 6 carbons in length, but the hexoketose has 3 chiral centers, and the hexoaldose is going to have 4. Okay. Let me get out of here. So when we talk about stereoisomers, the number of different optically and sterically different compounds is a function of the number of chiral centers. It is equal to 2 to the n, where n represents the number of chiral centers. So for a hexoaldose with 4 chiral centers, then they're going to be 16 different stereoisomers. For our hexoketoses, they're going to be 2 to the 3rd, or 8. So this is our aldose, well, specifically the hex 6 hexoaldose. And 8 for the keto aldose. Okay. So they're in talking about sugars, rather than giving you complicated naming for each of the sugars, what they have done instead is, for the most of them, they have given them different names. Okay? So what that means is that one of these hexoaldoses is called glucose. Another is called mannose. Another is called galactose. And the same is true. One of these is called fructose, but there are 7 other hexoketoses, each of which is described differently, most often by a change in its name. Okay, I think we'll leave it here. We'll come back and talk about some more terminology and talking about sugars. Yes, please.
Is hexagonal the same thing as all the hexos?
Yes, yes. If you have a preference, that's great. So. All right, let me begin a story. So many years ago, I was a college student, and this was during the Vietnam War era. And what that meant for people of your age was that at the age of 16, if you were a guy, that you were required by law to register with your draft board. Each community had its own draft board office, and you would go in and you'd give them all of your relevant information, and they would then put you into their system to keep track of you thereafter. And people were being called up to serve in the military through this draft. What that meant was that it wasn't a volunteer army. You were actually called up rather randomly and asked to serve in the military. Now, as I left high school and went on to college, I had what was called a student deferment. That is to say, as long as I stayed in good standing, carried a full load in college, I was not eligible for active duty. I had this student deferment. And so I went along, and I knew that the war was going on. I knew that people were getting drafted and having to serve in the military, but myself and fellow students enjoyed this delay, this deferment in our time of service to a later time. So during the first part of my senior year, I had applied for and been accepted to graduate school. And I assumed that if I went to graduate school and carried a full load and remained in good standing, that I would continue to have my student deferment. And I thought this was great. I'd be able to finish my education, then serve, and then come back and make use of my degree. So during my senior year, sort of in the middle somewhere, I decided that I would contact my draft board and tell them that I had been accepted to grad school, and to make sure that they continued my student deferment. So I called and I explained my situation and said, so, could you please make sure that my deferment is continued? And they say, no. So your student deferment is only for undergraduate education. We don't care if you're accepted to med school or engineering school or whatever. We don't care. Within a couple of weeks of your graduation from college, you will be in uniform and be on your way to Vietnam. I said, oh, thank you. I didn't realize that. That's interesting. So anyway, at that point, I realized why all of my friends were looking at National Guard units and reserve units, Army Reserve, Naval Reserve, so forth. Because this was a way of fulfilling your obligation to your country. And some of these units were being called up for active duty, but it allowed you, in most cases, to be able to continue schooling and not have to leave everything and go off for military training and military duty. So I began to look at all of these different reserve and National Guard units in my area, and all of them had waiting lists that were 2 to 3 years long. they had a pretty good idea of the turnover in their unit. They knew who was going to fulfill their time in service, and they assumed that a fair number of those would not re-up, would not continue. And so they knew that maybe they would need 10 people over the next year. Well, they had 30 or 35 people on their waiting list. And that meant that if you were lucky, 3 years from now, you might be able to fill a slot in one of these units. So my prospects of going into the National Guard or a reserve unit were very slim. We'll leave it there. Thank you very much. Quiz is due today. I'm going to have my review, my office hours here in about 20 minutes. Please join.